ID the Future, a podcast about evolution and intelligent design.
Welcome to ID the Future. I'm your host, Andrew McDiarmid. Today, I'm reading for you an essay by Yale University Professor of Computer Science, David Gelernter, published in the spring 2019 issue of the Claremont Review of Books. It's titled, Giving Up Darwin, A Fond Farewell to a Brilliant and Beautiful Theory. Gelernter's essay raised many an eyebrow when it was published 5 years ago, and for good reason. Here's an accomplished writer, artist, and thinker, famed both for his specific scientific expertise and for his cultural, political, and historical reflections, publicly declaring that Darwin's brilliant and beautiful scientific theory has now been overtaken by science. Gelernter credits reading Stephen Meyer's book Darwin's Doubt as the primary cause of his rejecting neo-Darwinian evolution, calling Darwin's Doubt one of the most important books in a generation. Few open-minded people will finish it with their faith in Darwin intact. Gelernter is no creationist, nor is he a proponent of intelligent design, but he is one of a growing number of scientists and thinkers bold enough to look beyond a Darwinian paradigm to explain the development of life on Earth. I'm reading Gelerner's essay to mark the 5-year anniversary of its publication. If you've read it, this will likely be a good refresher on Gelerner's arguments. If you haven't read it yet, by all means enjoy. We'll also post a link to the article in the show notes for this episode at idthefuture.com. Giving Up Darwin, A Fond Farewell to a Brilliant and Beautiful Theory, by David Gelernter. Darwinian evolution is a brilliant and beautiful scientific theory. Once, it was a daring guess. Today, it is basic to the credo that defines the modern worldview. Accepting the theory as settled science, no more subject to debate than the Earth being round, or the sky blue, or force being mass times acceleration, certifies that you are devoutly orthodox in your scientific views, which in turn is an essential first step towards being taken seriously in any part of modern intellectual life. But what if Darwin was wrong? Like so many others, I grew up with Darwin's theory, and had always believed it was true. I'd heard doubts over the years from well-informed, sometimes brilliant people, but I had my hands full cultivating my garden, and it was easier to let biology take care of itself. But in recent years, reading and discussion have shut that road down for good. This is sad. It is no victory of any sort for religion. It is a defeat for human ingenuity. It means one less beautiful idea in our world, and one more hugely difficult and important problem back on mankind's to-do list. But we each need to make our peace with the facts, and not try to make life on Earth simpler than it really is. Charles Darwin explained monumental change by making one basic assumption: all life forms descend from a common ancestor, and adding two simple processes anyone can understand: random heritable variation and natural selection. Out of these simple ingredients, conceived to be operating blindly over hundreds of millions of years, he conjured up change that seems like the deliberate unfolding of a grand plan, designed and carried out with superhuman genius. Could nature really have pulled out of its hat the invention of life, of increasingly sophisticated life forms, and ultimately the unique in the cosmos, so far as we know, human mind, given no strategy but trial and error, the mindless accumulation of small changes? It's an astounding idea. Yet Darwin's brilliant and lovely theory explains how it could have happened. Its beauty is important. Beauty is often a telltale sign of truth. Beauty is our guide to the intellectual universe, walking beside us through the uncharted wilderness, pointing us in the right direction, keeping us on track, most of the time. Demolishing a worldview. There's no reason to doubt that Darwin successfully explained the small adjustments by which an organism adapts to local circumstances. Changes to fur density, or wing style, or beak shape. Yet there are many reasons to doubt whether he can answer the hard questions and explain the big picture. Not the fine-tuning of existing species, but the emergence of new ones. The origin of species is exactly what Darwin cannot explain. Stephen Meyer's thoughtful and meticulous Darwin's Doubt, published in 2013, convinced me that Darwin has failed. He cannot answer the big question. Two other books are also essential. The Deniable Darwin and Other Essays, published in 2009 by David Berlinski, and Debating Darwin's Doubt, a 2015 anthology edited by David Klinghoffer, which collects some of the arguments Mayer's book stirred up. These three form a fateful battle group that most people would rather ignore. Bringing to bear the work of many dozen scientists over many decades, Mayer, who after a stint as a geophysicist in Dallas earned a PhD in the history and philosophy of science from Cambridge and now directs the Discovery Institute's Center for Science and Culture, disassembles the theory of evolution, piece by piece. Darwin's Doubt is one of the most important books in a generation. Few open-minded people will finish it with their faith in Darwin intact. Mayer doesn't only demolish Darwin, he defends a replacement theory, intelligent design, or ID. Although I can't accept intelligent design as Mayer presents it, he does show that it's a plain case of the emperor's new clothes. It says aloud what anyone who ponders biology must think at some point while sifting possible answers to hard questions. Intelligent design, as Mayer explains it, never uses religious arguments, draws religious conclusions, or refers to religion in any way. It does underline an obvious but important truth. Darwin's mission was exactly to explain the flagrant appearance of design in nature. The religion is all on the other side. Mayer and other proponents of ID are the dispassionate intellectuals making orderly scientific arguments. Some ID haters have shown themselves willing to use any argument, fair or not, true or not, ad hominem or not, to keep this dangerous idea locked in a box forever. They remind us of the extent to which Darwinism is no longer just a scientific theory, but the basis of a worldview, and an emergency replacement religion for the many troubled souls who need one. As for biblical religion, it forces its way into the discussion, although Mayer didn't invite it, and neither did Darwin. Some have always been bothered by the harm Darwin is said to have done religion. His theory has been thought by some naives, fundamentalists as well as intellectuals, to have shown or alleged that the Bible is wrong and Judeo-Christian religion bunk. But this view assumes a childishly primitive reading of Scripture. Anyone can see that there are two different creation stories in Genesis, one based on 7 days, the other on the Garden of Eden. When the Bible gives us two different versions of one story, it stands to reason that the facts on which they disagree are without basic religious significance. The facts on which they agree are the ones that matter. God created the universe and put man there for a reason. Darwin has nothing to say on these or any other key religious issues. Fundamentalists and intellectuals might go on arguing these things forever, but normal people will want to come to grips with Mayer and the downfall of a beautiful idea. I'll mention several of his arguments, one of them in just a bit of detail. This is one of the most important intellectual issues of modern times, and every thinking person has the right and duty to judge for himself. Looking for evidence. Darwin himself had reservations about his theory, shared by some of the most important biologists of his time, and the problems that worried him have only grown more substantial over the decades. In the famous Cambrian explosion of around 500 million years ago, a striking variety of new organisms, including the first ever animals, pop up suddenly in the fossil record over a mere 70-odd million years. This great outburst followed many hundreds of millions of years of slow growth and scanty fossils, mainly of single-celled organisms, dating back to the origins of life, roughly 3.5 billion years ago. Darwin's theory predicts that new life forms evolve gradually from old ones in a constantly branching, spreading tree of life. Those brave new Cambrian creatures must therefore have had pre-Cambrian predecessors, similar but not quite as fancy and sophisticated. They could not have all blown out suddenly, like a bunch of geysers. Each must have had a closely related predecessor, which must have had its own predecessors. Darwinian evolution is gradual, step by step. All those predecessors must have come together, further back, into a series of branches leading down to the long-ago trunk. But those predecessors of the Cambrian creatures are missing. Darwin himself was disturbed by their absence from the fossil record. He believed they would turn up eventually. Some of his contemporaries, such as the eminent Harvard biologist Louis Agassiz, held that the fossil record was clear enough already and showed that Darwin's theory was wrong. Perhaps only a few sites had been searched for fossils, but they had been searched straight down. The Cambrian explosion had been unearthed, and beneath those Cambrian creatures, their Precambrian predecessors should have been waiting, and weren't. In fact, the fossil record as a whole lacked the upward branching structure Darwin predicted. The trunk was supposed to branch into many different species, each species giving rise to many genera, and towards the top of the tree you would find so much diversity that you could distinguish separate phyla, the large divisions, sponges, mosses, mollusks, chordates, and so on, that comprise the kingdom of animals, plants, and several others. Take your pick. But as Berlinski points out, the fossil record shows the opposite. Representatives of separate phyla appearing first, followed by lower-level diversification on those basic themes. In general, most species enter the evolutionary order fully formed and then depart unchanged. The incremental development of new species is largely not there. Those missing Precambrian organisms have still not turned up. Although fossils are subject to interpretation, and some biologists place Precambrian life forms closer than others to the newfangled Cambrian creatures. Some researchers have guessed that those missing Precambrian precursors were too small or too soft-bodied to have made good fossils. Meyer notes that fossil traces of ancient bacteria and single-celled algae have been discovered. Smallness per se doesn't mean that an organism can't leave fossil traces, although the existence of fossils depends on the surroundings in which the organism lived, and the history of the relevant rock during the ages since it died. The story is similar for soft-bodied organisms. Hard-bodied forms are more likely to be fossilized than soft-bodied ones, but many fossils of soft-bodied organisms and body parts do exist. Precambrian fossil deposits have been discovered in which tiny, soft-bodied embryo sponges are preserved, but no predecessors to the celebrity organisms of the Cambrian explosion. This sort of negative evidence can't ever be conclusive, but the ever-expanding fossil archives don't look good for Darwin, who made clear and concrete predictions that have so far been falsified, according to many reputable paleontologists anyway. When does the clock run out on those predictions? Never. But any thoughtful person must ask himself whether scientists today are looking for evidence that bears on Darwin, or looking to explain away evidence that contradicts him. There are some of each. Scientists are only human, and their thinking, like everyone else's, is colored by emotion. The advent of molecular biology. Darwin's main problem, however, is molecular biology. There was no such thing in his own time. We now see from inside what he could only see from outside, as if he had developed a theory of mobile phone evolution without knowing that there were computers and software inside, or what the digital revolution was all about. Under the circumstances, he did brilliantly. Biology in his time was for naturalists, not laboratory scientists. Dr. Dolittle was a naturalist. He's the hero of the wonderful children's books by Hugh Lofting, now unfortunately nearing extinction. The doctor loved animals and understood them, and had a sharp eye for all of nature, not too different from Wordsworth's or Goethe's. But the character of the field has changed, and it's not surprising that old theories don't necessarily still work. Darwin's theory is simple to grasp. Its simplicity is the heart of its brilliance and power. We all know that variation occurs naturally among individuals of the same type: white or black sheep, dove-gray versus off-white or pale beige pigeons, boring and sullen undergraduates versus charming, lissom ones. We all know that many variations have no effect on a creature's prospects, but some do. A sheep born with extra warm wool will presumably do better at surviving a rough Scottish winter than his normal wooled friends. Such a sheep would be more likely than normal sheep to live long enough to mate and pass on its superior trait to the next generation. Over millions of years, small, good-for-survival variations accumulate, and eventually, says Darwin, you have a brand new species. The same mechanism naturally favors genes that are right for the local environment. Warm wool in Scotland, light and comfortable wool for the tropics, other varieties for mountains and deserts. Thus, one species, your standard sheep, might eventually become 4 specialized ones. And thus, new species should develop from old in the upward branching tree pattern that Darwin described. The advent of molecular biology made it possible to transform Darwinism into neo-Darwinism. The new version explains, it doesn't merely cite, natural variation as the consequence of random change or mutation to the genetic information within cells that deal with reproduction. Those cells can pass genetic change onward to the next generation, thus changing, potentially, the future of the species and not just one individual's career. The engine that powers neo-Darwinian evolution is pure chance and lots of time. By filling in the details of cellular life, molecular biology makes it possible to estimate the power of that simple mechanism. But what does generating new forms of life entail? Many biologists agree that generating a new shape of protein is the essence of it. Only if neo-Darwinian evolution is creative enough to do that is it capable of creating new life forms and pushing evolution forward. Proteins are the special ops forces, or maybe the Marines, of living cells, except that they are common instead of rare. They do all the heavy lifting, all the tricky and critical assignments in a dazzling range of roles. Proteins, called enzymes, catalyze all sorts of reactions and drive cellular metabolism. Other proteins, such as collagen, give cells shape and structure, like tent poles, but in far more shapes. Nerve function, muscle function, and photosynthesis are all driven by proteins. And in doing these jobs and many others, the actual 3D shape of the protein molecule is important. So is the simple neo-Darwinian mechanism up to the task? Are random mutation plus natural selection sufficient to create new protein shapes? Mutations. How to make proteins is our first question. Proteins are chains, linear sequences of atom groups, each bonded to the next. A protein molecule is based on a chain of amino acids. 150 elements is a modest-sized chain. The average is 250. Each link is chosen, ordinarily, from one of 20 amino acids. A chain of amino acids is a polypeptide, peptide being the type of chemical bond that joins one amino acid to the next. But this chain is only the starting point. Chemical forces among the links make parts of the chain twist themselves into helices. Others straighten out, and then sometimes jackknife repeatedly, like a carpenter's rule, into flat sheets. Then the whole assemblage folds itself up like a complex sheet of origami paper, and the actual 3D shape of the resulting molecule is, as I've said, important. Imagine a 150-element protein as a chain of 150 beads, each bead chosen from 20 varieties. But only certain chains will work. Only certain bead combinations will form themselves into stable, useful, well-shaped proteins. So how hard is it to build a useful, well-shaped protein? Can you throw a bunch of amino acids together and assume that you'll get something good? Or must you choose each element of the chain with painstaking care? It happens to be very hard to choose the right beads. Inventing a new protein means inventing a new gene. Enter, finally, genes, DNA, etc., with suitable fanfare. Genes spell out the links of a protein chain, amino acid by amino acid. Each gene is a segment of DNA, the world's most admired macromolecule. DNA, of course, is the famous double helix or spiral staircase, where each step is a pair of nucleotides. As you read the nucleotides along one edge of the staircase, sitting on one step and bumping your way downwards to the next and the next, each group of 3 nucleotides along the way specifies an amino acid. Each 3-nucleotide group is a codon, and the correspondence between codons and amino acids is the genetic code. The 4 nucleotides in DNA are abbreviated T, A, C, and G, and you can look up the code in a high school textbook. TTA and TTC stand for phenylalanine, TCT for serine, and so on. Your task is to invent a new gene by mutation, by the accidental change of one codon to a different codon. You have 2 possible starting points for this attempt. You could mutate an existing gene, or mutate gibberish. You have a choice because DNA actually consists of valid genes separated by long sequences of nonsense. Most biologists think that the nonsense sequences are the main source of new genes. If you tinker with a valid gene, you will almost certainly make it worse, to the point where its protein misfires and endangers or kills its organism, long before you start making it better. The gibberish sequences, on the other hand, sit on the sidelines without making proteins, and you can mutate them, so far as we know, without endangering anything. The mutated sequence can then be passed on to the next generation, where it can be mutated again. Thus, mutations can accumulate on the sidelines without affecting the organism. But if you mutate your way to an actual valid new gene, your new gene can create a new protein and thereby potentially play a role in evolution. Mutations themselves enter the picture when DNA splits in half down the center of the staircase, thereby allowing the enclosed cell to split in half and the encompassing organism to grow. Each half staircase summons a matching set of nucleotides from the surrounding chemical soup. 2 complete new DNA molecules emerge. A mistake in this elegant replication process, the wrong nucleotide answering the call, a nucleotide typo, yields a mutation, either to a valid blueprint or a stretch of gibberish. Building a better protein. Now at last we're ready to take Darwin out for a test drive. Starting with 150 links of gibberish, what are the chances that we can mutate our way to a useful new shape of protein? We can ask basically the same question in a more manageable way. What are the chances that a random 150 link sequence will create such a protein? Nonsense sequences are essentially random. Mutations are random. Make random changes to a random sequence and you get another random sequence. So close your eyes, make 150 random choices from your 20 bead boxes and string up your beads in the order in which you chose them. What are the odds that you'll come up with a useful new protein? It's easy to see that the total number of possible sequences is immense. It's easy to believe, although non-chemists must take their colleagues' word for it, that the subset of useful sequences, sequences that create real, usable proteins, is, in comparison, tiny. But we must know how immense and how tiny. The total count of possible 150 link chains, where each link is chosen separately from 20 amino acids, is 20 to the 150th power. In other words, many. 20 to the 150 roughly equals 10 to the 195th, and there are only 10 to the 80th atoms in the universe. What proportion of these many polypeptides are useful proteins? Douglas Axe did a series of experiments to estimate how many 150 long chains are capable of stable folds, of reaching the final step in the protein creation process, the folding, and of holding their shapes long enough to be useful. Axe, by the way, is a distinguished biologist with 5-star breeding. He was a graduate student at Caltech, then joined the Center for Protein Engineering at Cambridge. The biologists whose work Meyer discusses are mainly first-rate establishment scientists. Axe estimated that of all 150 link amino acid sequences, 1 in 10 to the 74th will be capable of folding into a stable protein. To say that your chances are 1 in 10 to the 74th is no different in practice from saying that they're 0. It's not surprising that your chances of hitting a stable protein that performs some useful function and might therefore play a part in evolution are even smaller. Axe puts them at 1 in 10 to the 77th. In other words, immense is so big and tiny is so small that neo-Darwinian evolution is, so far, a dead loss. Try to mutate your way from 150 links of gibberish to a working, useful protein, and you are guaranteed to fail. Try it with 10 mutations, 1,000, 1,000,000. You fail. The odds bury you. It can't be done. A bad bet. But neo-Darwinianism understands that mutations are rare, and successful ones even scarcer. To balance that out, there are many organisms and a staggering immensity of time. Your chances of winning might be infinitesimal, but if you play the game often enough, you win in the end, right? After all, it works for Powerball. Do the numbers balance out? Is neo-Darwinian evolution plausible after all? Axe reasoned as follows. Consider the whole history of living things, the entire group of every living organism ever. It is dominated numerically by bacteria. All other organisms, from tangerine trees to coral polyps, are only a footnote. Suppose, then, that every bacterium that has ever lived contributes one mutation before its demise to the history of life. This is a generous assumption. Most bacteria pass on their genetic information unchanged, unmutated. Mutations are the exception. In any case, there have evidently been, in the whole history of life, around 10 to the 40th bacteria, yielding around 10 to the 40th mutations under Axe's assumptions. That's a very large number of chances at any game. But given that the odds each time are 1 in 10 to the 77th against, it is not large enough. The odds against blind Darwinian chance having turned up even one mutation with the potential to push evolution forward are 10 to the 40th times 1 in 10 to the 77th. 10 to the 40th tries, where your odds of success each time are 1 in 10 to the 77th, which equals 1 in 10 to the 37th. In practical terms, those odds are still zero. Zero odds of producing a single promising mutation in the whole history of life. Darwin loses. His idea is still perfectly reasonable in the abstract, but concretely, he is overwhelmed by numbers he couldn't possibly have foreseen. The ridiculously large number of amino acid chains relative to number of useful proteins. Those numbers transcend the details of any particular set of estimates. The obvious fact is that genes, in storing blueprints for the proteins that form the basis of cellular life, encode an awe-inspiring amount of information. You don't turn up a useful protein merely by doodling on the back of an envelope, any more than you write a Mozart aria by assembling 3 sheets of staff paper and scattering notes around. Profound biochemical knowledge is somehow, in some sense, captured in every description of a working protein. Where on earth did it all come from? Neo-Darwinianism says that nature simply rolls the dice, and if something useful emerges, great. Otherwise, try again. But useful sequences are so gigantically rare that this answer simply won't work. Studies of the sort Meyer discusses show that neo-Darwinism is the quintessence of a bad bet. The Great Darwinian Paradox. There are many other problems besides proteins. One of the most basic, and the last I'll mention here, calls into question the whole idea of gene mutations driving macroevolution, the emergence of new forms of organism, versus mere variation on existing forms. To help create a brand new form of organism, a mutation must affect a gene that does its job early and controls the expression of other genes that come into play later on as the organism grows. But mutations to these early-acting strategic genes, which create the big body plan changes required by macroevolution, seem to be invariably fatal. They kill off the organism long before it can reproduce. This is common sense. Severely deformed creatures don't ever seem fated to lead the way to glorious new forms of life. Instead, they die young. Evidently, there are a total of no examples in the literature of mutations that affect early development and the body plan as a whole and are not fatal. The German geneticists Christian Nusslein-Volhart and Erich Wieschaus won the Nobel Prize in 1995 for the Heidelberg screen, an exhaustive investigation of every observable or inducible mutation of Drosophila melanogaster, the same patient, long-suffering fruit fly I meddled with relentlessly in an undergraduate genetics lab in the 1970s. We think we've hit all the genes required to specify the body plan of Drosophila, said Wieschaus in answering a question after a talk. Not one, he continued, is promising as raw materials for macroevolution, because mutations in them all killed off the fly long before it could mate. If an exhaustive search rules out every last plausible gene as a candidate for large-scale Drosophila evolution, where does that leave Darwin? Wieschaus continues, what are, or what would be, the right mutations for major evolutionary change? And we don't know the answer to that. There is a general principle here, similar to the earlier principle that the number of useless polypeptides crushes the number of useful ones. The Georgia Tech geneticist John F. MacDonald calls this one a great Darwinian paradox. Meyer explains, genes that are obviously variable within natural populations seem to affect only minor aspects of form and function, while those genes that govern major changes, the very stuff of macroevolution, apparently do not vary or vary only to the detriment of the organism. The philosopher of biology Paul Nelson summarizes the body plan problem. Research on animal development and macroevolution over the last 30 years, research done from within the neo-Darwinian framework, has shown that the neo-Darwinian explanation for the origin of new body plans is overwhelmingly likely to be false, and for reasons that Darwin himself would have understood. Darwin would easily have understood that minor mutations are common, but can't create significant evolutionary change. Major mutations are rare and fatal. It can hardly be surprising that the revolution in biological knowledge over the last half-century should call for a new understanding of the origin of species. Darwin's limits. Intelligent design, as Meyer describes it, is a simple and direct response to a specific event, the Cambrian explosion. The theory suggests that an intelligent cause intervened to create this extraordinary outburst. By intelligent, Meyer understands conscious. The theory suggests nothing more about the designer. But where is the evidence? To Meyer and other proponents, that is like asking, after you've come across a tree that is split vertically down the center and half burnt up, but where is the evidence of a lightning strike? The exceptional intricacy of living things and their elaborate mechanisms for fitting precisely into their natural surroundings seem to cry out for an intelligent designer long before molecular biology and biochemistry. Darwin's theory, after all, is an attempt to explain design without a designer, according to evolutionary biologist Francisco Ayala. An intelligent designer might seem more necessary than ever now that we understand so much cellular biology, and the impossibly long odds facing any attempt to design proteins by chance, or assemble the regulatory mechanisms that control the life cycle of a cell. Meyer doesn't reject Darwinian evolution. He only rejects it as a sufficient theory of life as we know it. He's made a painstaking investigation of Darwin's theory, and has rejected it for many good reasons that he has carefully explained. He didn't rush to embrace intelligent design, just the opposite. But the explosion of detailed, precise information that was necessary to build the brand new Cambrian organisms, and the fact that the information was encoded, represented symbolically in DNA nucleotides, suggests to Meyer that an intelligent designer must have been responsible. Our uniform experience of cause and effect shows that intelligent design is the only known cause of the origin of large amounts of functionally specified digital information, he writes. Digital is confusing here. It only means information represented by a sequence of symbols. Was the Cambrian explosion unique in some absolute sense, or was it the extreme endpoint of a spectrum? After all, there were infusions of new genetic information before and after. Meyer himself writes that the sudden appearance of the Cambrian animals was merely the most outstanding instance of a pattern of discontinuity that extends throughout the geologic column. It's not easy to decide whether something stands alone or at the far end of some spectrum. Consider Meyer's functionally specified digital information. Information intended for one specific purpose and spelled out in a sequence of symbols is a rare bird in nature. It's an outlier in the world of intelligence, too. We nearly always communicate in symbols that are used for many purposes. It's hard for us to confine any symbol system to a single purpose. Even digits are used to represent numbers of many kinds, to express order as well as magnitude, as names, like 2001: A Space Odyssey, or parts of English phrases, like second rate. A line of music can be heard in the head, hummed or sung, played on a zither, or performed by a large orchestra. Or it can serve as a single graphic symbol meaning music. But the genetic code is used to specify the structure of certain molecules only, albeit in a series of separate steps and information transfers within the cell. Nature, for its part, encodes information in many ways. Airborne scents are important to bees, butterflies, elephants seeking to mate, birds avoiding trouble, and untold other creatures. The scent is a symbol. It's not the scent that threatens the bird. Channels in sand dunes encode information about the passing breezes, and so on. There are endless examples, none approaching the sophistication and complexity of DNA coding. If Meyer were invoking a single intervention by an intelligent designer at the invention of life, or of consciousness, or rationality, or self-aware consciousness, the idea might seem more natural. But then we still haven't explained the Cambrian explosion. An intelligent designer who interferes repeatedly, on the other hand, poses an even harder problem of explaining why he chose to act when he did. Such a cause would necessarily have some sense of the big picture of life on Earth. What was his strategy? How did he manage to back himself into so many corners, wasting energy on so many doomed organisms? Granted, they might each have contributed genes to our common stockpile, but could hardly have done so in the most efficient way. What was his purpose? And why did he do such an awfully slipshod job? Why are we so disease-prone, heartbreak-prone, and so on? An intelligent designer makes perfect sense in the abstract. The real challenge is how to fit this designer into life as we know it. Intelligent design might well be the ultimate answer, but as a theory, it would seem to have a long way to go. A final challenge. I might, myself, expect to find the answer in a phenomenon that acts as if it were a new, and thus far, unknown force or field associated with consciousness. I'd expect complex biochemistry to be consistently biased in the direction that leads closer to consciousness, as gravitation biases motion towards massive objects. I have no evidence for this idea, it's just the way biology seems to work. Although Stephen Meyer's book is a landmark in the intellectual history of Darwinism, the theory will be with us for a long time, exerting enormous cultural force. Darwin is no Newton. Newton's physics survived Einstein, and will always survive, because it explains the cases that dominate all of space-time, except for the extreme ends of the spectrum, at the very smallest and largest scales. It's just these most important cases, the ones we see all around us, that Darwin cannot explain. Yet his theory does explain cases of real significance. And Darwin's intellectual daring will always be inspiring. The man will always be admired. He now poses a final challenge. Whether biology will rise to this last one as well as it did to the first, when his theory upset every apple cart, remains to be seen. How cleanly and quickly can the field get over Darwin and move on, with due allowance for every Darwinist's having to study all the evidence for himself? There is one of the most important questions facing science in the 21st century. That was an essay by Yale University polymath David Gelernter called Giving Up Darwin, a fond farewell to a brilliant and beautiful theory. It was originally published in the spring 2019 issue of the Claremont Review of Books. We'll include a link to the article in the show notes for this episode over at idthefuture.com. If you enjoyed Gelernter's arguments, forward this episode to a friend. For ID the Future, I'm Andrew McDiarmid. Thanks for listening.
Visit us at idthefuture.com and intelligentdesign.org. This program is copyright Discovery Institute and recorded by its Center for Science and Culture.